In Chapter 17, Segment 1, in Functional Perspectives on Learning and Memory, it defines learning as the process of acquiring new and relatively enduring information, behavior patterns, or abilities characterized by modifications of behavior as a result of practice, study, or experience. It also defines memory as the ability to retain information based on the same mental process of learning or encoding, retention across some interval of time, and retrieval or reactivation of the memory. It is also defined as the specific information that is stored in the brain. Memory can fall in many different ways, which shows that there are different forms of learning and memory and that multiple brain regions are involved. And there are several kinds of learning and memory. It says we cannot be sure that learning has occurred unless a memory can be obtained later. And many different kinds of brain damage caused by disease or accidents can produce highly specific types of memory impairment. For patient HM, the present vanished into oblivion. It defines amnesia as severe impairment of memory. Retrograde amnesia is difficulty in retrieving memories formed before the onset of amnesia. After an accident that damages the brain, people often have retrograde amnesia with regard to events that happened a few hours or days before the accident or even a year. Patient HM or Henry Molaison, a person who, because of damage to the medial temporal lobe structures, was unable to encode new decorative memories. Anterograde amnesia is the inability to form new memories beginning with the onset of a disorder. In Henry's case, most old memories remained intact, but he had difficulty recollecting any events after surgery. His disorder proved that short-term memory is different from long-term memory. Henry's surgery removed the amygdala, most of the hippocampus, and some surrounding cortex from both temporal lobes. The memory deficit seemed to come from loss of the medial temporal lobe, including the hippocampus. Other surgical patients who had the same kind of damage to the amygdala but less damage to the hippocampus did not exhibit comparable memory impairment. Bilateral hippocampal lesions in laboratory animals did not produce comparable memory deficits. When Henry was given a mirror tracing task, he improved over the 10 trials, but when he was asked if he recognized the task, he said no. Henry never recognized the task as something he had seen before, but his improved tracing showed evidence of memory in the form of motor skill. People with brain damage like Henry's have difficulty reproducing or recognizing pictures and spatial designs that are not recalled with verbal terms. Although such individuals have difficulty with specific content of verbal material, they can learn some kinds of information about verbal material. For example, people with several kinds of amnesia can learn the skill of reading mirror reverse text but show impaired learning of specific words. The important distinction is not between motor and verbal performances, but between two general categories of memory. The first is declarative memory, is what we usually think of memory. It's facts and information required by learning, and it is memory we are aware of accessing, which we can declare to others, the kind of memory impaired by Henry's surgery. It is also the type of memory we use to answer what questions, and thus is difficult to test in animals. The second is non-declarative memory, which is memory about perceptual or motor procedures. It is shown by performance rather than by conscious recollection, and is things that you learned by doing. It is also the type of memory we use for how problems and is often nonverbal. Medial temporal lobe structures are crucial for declarative memory. It says delayed non-matching to sample task is a test in which on each trial the participant must select a stimulus that they have not seen previously. For example, monkeys must identify which of the two objects was not seen previously with delays ranging from 8 seconds to 2 minutes. The important thing is that the animal does not reach for the item that previously had a treat under it, because in that case the monkey might unconsciously associate reward with that object even if the animal had no conscious recollection of it. Instead, the monkey declares that it remembers the object by reaching for the other object, the one that was not associated with the reward previously. Monkeys with extensive damage to the medial temporal lobe are severely impaired on this test, especially with the longer delays. Selective removal of specific parts in the medial temporal lobes in monkeys revealed that the amygdala was not crucial for performance on tests of declarative memory. Removal of the adjacent hippocampus significantly impaired performance on these tests, and the deficit was even more pronounced when the damage was paired with lesions of the nearby entorhinal and parahippocampal cortex, and much worse when lesions of perirhinal cortex were added. One modern formulation of declarative memory for facts and events takes the view that the two interwoven processes are involved: recollection of the item in the specific context in which it was presented, and familiarity with the features of the recalled item. Processing of contextual aspects of memory appears to depend especially on the parahippocampal cortex. The hippocampus acts as the final stage of convergence for adjacent regions of cortex, resulting in storage of declarative memories in the cortex. In general, the performance of experimental animals suggests that the hippocampus acts as the final stage of convergence for adjacent regions of cortex. People with specific hippocampal damage appear to have difficulties with recollection, while familiarity-based aspects of memory are spared. People with surgical lesions that include the perimordial cortex but not the hippocampus experience impaired familiarity with stimuli despite preserved recollection. And damage to the medial diencephalon can also cause amnesia. It says that patient NA was a person who was unable to encode new declarative memories because of damage to the dorsal thalamus and the mammillary bodies. An MRI showed that he had damage to several components of the diencephalon, thalamus and hypothalamus, that have connections to the hippocampus, especially the dorsal medial thalamus and both mammillary bodies, as well as probable damage to the mammothalamic tract. Mammillary bodies are one pair of nuclei at the base of the brain that connect the hippocampus and play a role in memory. Patient NA shows normal short-term memory but is impaired in forming declarative but not declarative procedural long-term memories. Similarity between NA and HM suggests that the medial temporal region damaged in Henry's brain and the midline diencephalic region damaged in NA are normally parts of a larger memory system. Studies of people with surgical lesions of the fornix, which interconnects the hippocampus and mammillary bodies, confirm and extend this idea as such individuals experience selective difficulty forming new declarative memories. Korsakoff syndrome is a memory disorder relating to a thiamine deficiency that is generally associated with chronic alcoholism. These people frequently deny that anything is wrong with them and also tend to confabulate things. Confabulate is to fill in a gap in memory with falsification. It often occurs in Korsakoff syndrome. Temporal lobe structures, including the hippocampus, are typically normal in people with Korsakoff syndrome, but the brains show shrunken, diseased mammillary bodies, as well as some damage in the dorsal medial thalamus. This damage is similar to that seen in NA. The mammillary bodies might serve as a processing system connecting to the medial temporal lobes to the thalamus via the mammothalamic tract and from there to other cortical sites. Damage to the basal frontal cortex, also found in people suffering from Korsakoff syndrome, probably causes a denial and confabulation that differentiates them from other people who have amnesia, such as Henry. The main cause of Korsakoff syndrome is lack of the vitamin thiamine, or B1. Alcoholics who obtain most of their calories from alcohol and neglect their diet often exhibit this deficiency. Treating them with thiamine can prevent further deterioration of memory functions but will not reverse the damage that has already been done. And the cortex is essential for long-term storage of memories. It says that patient KC was a person who sustained damage to the cortex that rendered him unable to form and retrieve new episodic memories, especially autobiographical memories. Upon his death, we learned his name was Ken Cochran. For example, he conversed easily and plays a good game of chess, but could not remember where he had learned to play chess or who taught him. Episodic memory is a memory of a particular incident or a particular time and place. Semantic memory is generalized memory, for instance, knowing the meaning of a word without knowing where and when you learned that word. Brain scans of Casey revealed extensive damage to the cerebral cortex and severe shrinkage of the hippocampus and nearby parahippocampal cortex. The bilateral hippocampal damage probably accounts for the anterior grade amnesia, but not selective loss of nearly his entire autobiographical memory, because other people with damage restricted to the medial temporal lobe lack this symptom. A group of 11 people with extreme autobiographical memory were found to have larger temporal lobes, including a larger parahippocampal gyrus than controls. In two different forms of non-declarative memory involved in different brain regions, it says that two different kinds of declarative memory, the semantic and episodic, and there are several forms of non-declarative memory that depend on different neural systems. In specific functions that depend on different forms of non-declarative memory, it says non-declarative memory shape our abilities and responses to the world around us in ways that are difficult to express but no less crucial for survival. Virtually every single species of the animal kingdom seems capable of some sort of non-declarative memory, even if very simple, indicating that it's an ancient evolutionary adaptation. In skill memory, it defines skill learning as learning to perform a task that requires motor coordination. Participants learn how to perform a challenging task by practicing it over and over and over again. For example, learning to read mirror reverse text and mirror tracing. Imaging studies have investigated learning and memory for different skill categories, especially sensory motor skills, perceptual skills, and cognitive skills. All three kinds of skill learning are impaired in people with damage to the basal ganglia. Damage to other brain regions, especially the motor cortex and cerebellum, also affects any aspects of these skills. Neuroimaging studies in healthy people confirm that the basal ganglia, cerebellum, and motor cortex are important for sensory motor skill learning. For example, learning specific sequences of finger movements is associated with selective activation of motor cortex in the basal ganglia, with the cerebellum possibly providing error connection during learning. In priming, it says that a change in the processing of stimulus, usually a word or picture as a result of prior exposure to the same stimulus or related stimuli, is referred to as priming. Priming is also called repetition priming. It is a phenomenon by which exposure to a stimulus facilitates subsequent responses to the same or similar stimulus. For example, if a person is shown the word stamp in a list and is later asked to complete the word stem STA, he or she is more likely to reply stamp than a person who has not been exposed to that word. Priming does not require declarative memory of the stimulus. Researchers have found that priming is not impaired by damage to the basal ganglia, and in functional imaging studies, perceptual priming has been related to reduced activity in bilateral occipitotemporal cortex, presumably because the priming makes the task easier. Conceptual priming was associated with reduced activation of the left frontal cortex, and components of the medial temporal lobe memory system, notably the perirhinal cortex and hippocampus, are also involved in some forms of non-declarative memory, including priming.